This report offers a comparative analysis of three major drug discovery approaches, namely structure-based drug discovery (SBDD), phenotypic-based drug discovery (PDD), and middle target signaling drug discovery (MTDD). We examine their scientific principles, strategic considerations, and intellectual property (IP) implications, providing a comprehensive view for researchers and innovators in the field. Structure-based drug discovery (SBDD) utilizes the three-dimensional structure of a target, usually a protein, to design a small molecule that can bind to it with high affinity and specificity. It is a rational approach that uses techniques like X-ray crystallography and cryo-electron microscopy to determine the structure of the target, and then employs structure-based design principles to develop ligands that fit into the target's active site. Phenotypic-based drug discovery (PDD). PDD, on the other hand, is a target-agnostic approach that begins with a screen for a desired phenotype, such as a change in cell morphology or a reduction in disease-related symptoms. It does not rely on prior knowledge of the target and can uncover novel mechanisms of action more. For instance, PDD has led to the discovery of drugs like Artemisinin for malaria and 𝗘𝗫𝗣𝗅 for cancer. Middle target signaling drug discovery (MTDD) focuses on key regulatory nodes or middle targets within signaling networks rather than the pathway's start or end. It involves identifying and modulating signaling components such as pseudokinase domains or scaffolding proteins to restore normal cellular function. MTDD has led to successful drugs like Venetoclax for cancer and 𝗑𝗫𝗣𝗅𝗘𝗏 for autoimmune disorders. First-in-class versus best-in-class. First-in-class drugs introduce a novel mechanism of action and typically target a previously unexploited biological pathway. Best-in-class drugs, on the other hand, improve upon existing drugs by offering better pharmacokinetics, safety, or selectivity. SBDD is often employed in best-in-class efforts. Real-world examples of success are WS. We present the top three are WS for each strategy. For SBDD, Imatinib, 𝗬𝗓𝗓𝗡, and sotorasib. For PDD, Artemisinin, 𝗘𝗫𝗣𝗅𝗏, and fingolimod. And for MTDD, Venetoclax, 𝗑𝗫𝗣𝗅𝗘𝗏, and Navitoclax. Comparative overview. SBDD is best suited when the biological target is known and structurally characterized. It offers medium discovery speed and relatively lower innovation risk. PDD, while more exploratory, is slower and riskier but more likely to produce first-in-class drugs. MTDD offers high innovation potential and medium discovery speed. IP strategy summary. SBDD focus on structural scaffold claims, active site binding specificity, and chemical analogs. Use crystallographic evidence to support patent applications. PDD emphasize phenotypic endpoints, therapeutic use claims, and later-stage MOA patents to broaden IP scope. Use of cell models and disease-specific screening data enhances defensibility. MTDD protect intervention strategies within signaling networks, including pseudokinase domains and adapter complexes. Pathway modulation and systems biology integration are key for broad utility claims. Strategic recommendations hybridize SBDD and PDD where possible, using phenotypic screening for lead identification and structural insights for optimization. Employ MTDD in indications characterized by complex signaling pathology, such as immuno-oncology and chronic inflammatory diseases. Early provisional patent filings can help secure IP positions across structure, function, and network interaction claims. By integrating computational biology, high-throughput screening, AI tools, and robust translational development, organizations can leverage these diverse discovery paradigms to deliver first-in-class therapies to patients in need. This report is a product of extensive research and analysis. If you found it valuable, please stay tuned for more insightful content. Your engagement helps us continue delivering quality research-driven reports.